Okay, so as I talked about this and the ability to modulate this up or down, that's one of our interests. We have actually individuals at Hopkins, I'll just mention their names. David Ginty has been recently working in this area of touch and the cells responsible and what their aspects of touch. Jinzhong Dang has been very interested in itch, which is everything from a relatively simple kind of itch phenomenon to chronic itch. And one of the major mechanisms that induces itch is actually the taking of drugs. So many drugs that individuals take actually induce quite severe itch. In fact, some cases so severe that those individuals discontinue use. I think one that Jinzhong talks about is chloroquine, involved in protection against malaria. And so he's been interested in the mechanisms by which we specifically detect itch and how itch is different than pain, even though all of these involve peripheral, at least initially involve peripheral mechanosensation. And so he's been interested in that phenomenon. You're not going to hear from him today, but then Mike Caterine has spent most of his career at Hopkins actually studying a variety of aspects of pain at that level. I'll just tell you, just to reemphasize this a little bit more, that there are molecular links between these sensory processes. And Mike Caterine, and work you'll hear a lot about today, was the pioneer in the recognition of the mechanism of thermal sensation and pain. And so I'm not going to say any more about that. What was recognized is that sensation, at least a component of that sensation, and related sensations derive from a very large family of proteins called tryp channels, originally identified by Craig Montell at Hopkins. And a variety of individuals have recognized that in mammals that these are not only involved in thermal sensation, but in other taste modalities, and they probably regulate some aspects of these sensory systems as well. So we're beginning to learn, not just from the nature of the organization, but even at the molecular level, how particular kinds of channels engage and participate in various pathways. I mentioned already that the senses are target of an environmental assault. They lie at the interface between our inner and outer worlds, and therefore they're subject to damage. As I said, you'll hear from Paul in just a moment about hearing pain, obviously one area that's very important is hearing loss, and we're interested in the mechanism there. Burn and itch, olfactory loss I talked about, and light-induced damage. Jeremy Nathans in the center has been interested in mechanisms of light and non-light-induced damage and diseases of the retina. So why are we interested in sensory biology? For a variety of different reasons. First, strategies that might allow us to modulate either up or down sensory perception, and you can hear a little bit more about that today. How we'd like to understand how the organization and interaction of cells in these tissues is organized as a paradigm for the more general process of moving information between cells in an organ. Secondly, actually work in my laboratory, but several other laboratories are interested in how specialized organelles are formed in sensory systems. We work on cilia, and cilia are shared in the olfactory system. They're very important in the chemosensation. In the auditory system for organizing the hair bundles essential for hearing, as well as in many of the other neurons of sensory systems. And then as a more general paradigm for other diseases. And then finally, we've been broadly interested in the processes of how neurons get damaged, how they are repaired, and particularly in the capacity for regeneration, whether that be the olfactory system where it's very robust, or in the auditory system where it is extremely modest, but of potential great clinical importance. So with that, I think I can give you a little perspective about at least the way we think about sensory systems in general. You're going to hear about pain, and that pain fits in with a broader spectrum of pleasant to quite noxious with things in the middle, and we can discriminate. They're not a simple spectrum because we can discriminate those, and they serve important things, not just in our ability to get in information, but to protect us from potentially dangerous things. And with that, I think I'll turn it over to Paul, who will begin to focus a little bit more, and then by the time we get to Mike, you'll get to really begin to hear the nuts and bolts of what we would call classic pain.